Hi. So, this is specifically for looking at the raw data for the sequencing. More specifically, if you find a paper where the data is deposited in the Chinese government site, then this is the way you need to download it for the further processing and analysis. So let's look for a paper. For example, this is one of the paper. So this is published by Shi Li Hong, and it's published in 2021. The title is Hematopoietic Stem Cell Heterogeneity. It's linked to the initiation and therapeutic response of myeloproliferative neoplasms. Let's go to the star part. And look for the data and code availability. So if you look at in here, specifically the data, specifically the single cell RNA-seq part is submitted to the Chinese government National Genomics Data Center, and the number is HRA 000669. So let's search for National Genomics Center. Not from here. You can't find anything to the American government site, which is NCBI. So we need to go in here. Now, as we have specifically the number for this data set is in here. So we go for search. In the first part, if you see GSA, which stands for Genome Sequence Archive for human. So let's go through this record. You can see single cell analysis of MPN HSCs. And when you come on this website, we just specifically have this data set. And this is metadata. So if you just click, you can download it and see you had the total patient information of 15 patients. Those sequencing was done NC. Stands for normal donor, ET, this essential thrombocythemia. The patients, specifically the first 6, was newly diagnosed, and next 4 they are treated with the specific drugs. Either way, this is the 15 patient data we have. This is the metadata of this sequencing part. So now if we want to look at, this is the individual samples we have, and then these are the files which has the run accession number, the experiment, and the samples. And these are the FASTQ files formats. Now if you want to specifically download one file, you can always try, okay, I want to run these. There is a FTP option and HTTPS option, and if you click on them you can download it, but the speed of downloading is pretty low for such huge quantity of data, which is approximately 400 GB or 500 GB. It's a tough chase to download all of this data one by one. Each sample contains 2 of the FASTQ files. So they suggest just download the data with FileZilla client or Aspira command line when both of them actually support the FTP. You can do the Aspira command line or the FileZilla client. So now how to get the FileZilla? It's luckily it's already into the Ubuntu App Center. So let's type FileZilla. So you can see this supports the graphical FTP, FTPS and SFTP client. So we just install it. As it is installed, we simply open it, and then I hope this is big enough. In the host area, you write download.big.ac.cn. You don't need to fill up the username, password, port will be automatically taken. So this actually specifically helps you to connect with the National Genomics Data Center of Chinese government. So click on this quick connect. You can see it's connecting in here. Connection established, waiting for welcome message. Just click on OK. It is logging in, and it has logged in here. Now on the right side, specifically the remote site area, you will look for a specific GSA that we have seen earlier. If you remember, there was GSA for human. So we look for this specific GSA for human part. So this is GSA human. So we click on this, and now you can see there are specific HRA numbers. Let's go back to the paper that we were looking for. It has the GSA number, which is HRA 000669. So we go for HRA 000669. Now these are all the files. If you say 4645 204645, let's compare. (204645). So as we have done double confirmation, now it's the time to download it. You just can click on this and say okay, just download. What will happen if we do that? It will start downloading. You will see in here in queue, it will start queued and then start connecting and downloading. Once it continues, it will finish up your entire data, which is 43.9 GB downloading in here. If you have multiple files, you just select them and click on the download. It will be queued, and one after one they will be downloading. You don't need to do much. And after that, in the meantime, you must have selected like local set where you want to store the data. So if you have selected, okay, this is the folder where I am saving my data. So let's go. We go to the download, and you see this is 204645, and both files are already downloading. And that's the way you download the data when it's deposited to the Chinese government site, any sort of sequencing data. And I hope that helps. Thank you.